Okay, good morning. We are going to continue on chapter 4 today. For Monday, you might want to read the last couple sections of chapter 4 that we're going to discuss, 12 and 13. We have homework. It's not due today. It's due a week from today, and we'll be talking about some of those problems today, hopefully at the end of class if we have time. All right, so I want to talk a little bit about diffusion today. And first we're going to spend most of our time talking about sort of a macroscopic model of diffusion, but I want to look a little bit at what's happening at the microscopic level first. So imagine you had some molecules, and let's imagine there's sort of an artificial line here separating the two halves of the region, the left half and the right half. And on the left there's a lot of these molecules. These could be oxygen. We'll be talking about oxygen a lot. And on the right, there is smaller numbers. So it's a concentration gradient. Now, molecules are moving around randomly in all directions because it has thermal motion. So you got some moving this way, some moving this way. These all have kind of random motion. And you see that some of them are crossing this line and going from the left side to the right side. Now, there are also molecules over on this side that have their own random motion. And some of them move from right to left. But, on the whole, since there's more molecules over here and the motion's completely random, there's going to be more moving from left to right than right to left. So, on the whole, it's going to seem like molecules are moving from the region of high concentration to low concentration. And that's really what's happening with diffusion, is microscopically the molecules are just randomly moving around, but the probability is that there's going to be more molecules going towards the region of lower concentration. So that's the way you can think about diffusion at a sort of microscopic scale. At a macroscopic scale, we're going to define some variables. We're going to define the concentration, which is just going to be the concentration. And that will be the number of molecules, or chemists would say moles, per unit volume. Meters cubed. Your chemist might say liters. So we'll be talking about concentration. It'll be one of the main variables when discussing diffusion. But we're also going to introduce this idea like we had last class when we were talking about heat. We're going to introduce a molecular current density. So J will be a molecular current density. And it's going to be really the number of molecules per unit area per unit time. So its units are molecules per unit area, meters squared, per unit time, second. And these are the two main quantities we're going to talk about. And then we're going to come up with an idea that says that the molecular current density, that's how many of these molecules are moving, it's going to be going down the concentration gradient. So we're going to come up with a result that looks a lot like what we saw for heat. It's going to say J is equal to, well, we're going to get a minus because it goes, in this case, if this was the positive x direction, you'd be going mainly from left to right, and that would be going down a concentration gradient. The gradient would be going really larger concentrations here, smaller concentrations here, so the derivative, dc dx, would be going down, yet the current is going to the right. You need the minus sign. And it's going to be, leave a little space, it's going to be proportional to the concentration gradient. This is very similar to what we derived last class for the temperature gradient. And the constant in here is going to be the diffusion constant. And that will be one of our main constants when we discuss diffusion. Okay, now, what are the units of the diffusion constant? Well, it's going to give you, on this side, molecules per meter squared second. And that's going to equal D, and then the concentration gradient is going to be molecules per meters cubed, but there's a derivative, so you get 1 over meters from the derivative. And so you can work out the molecules cancel. This will be meters to the fourth, which will come over here. D is going to end up giving meters to the fourth over meters squared. That's meters squared per second. Those are the units for the diffusion constant. It's easily, sometimes I get it mixed up with acceleration. Acceleration is meters per second squared. Diffusion constant, meters squared per second. Okay. Now the next thing I want to do is something that's very similar to last class, where we're going to look at sort of a continuity equation to see if we can tell how these molecules are changing with space. So let's look at either a piece of tissue or just some region of space with an area A and a thickness delta X. So if you follow this last class, it's going to be the same type of argument. You're going to have a current coming in. So molecules are going to be entering from the left, and they're going to be leaving, possibly a different number, from the right. So how many we want to figure out is how many molecules are building up, because we could have, if we had more coming in from the left than the right, that just means molecules are building up in this region. So it's kind of like if you had more cars leaving Rochester Hills and then driving down to Troy, then that means cars leaving Rochester Hills must be stopping in somewhere between the two cities and piling up there, and their concentration's getting bigger. So how many molecules are coming in from the left? What did we say that was? Per unit time. What is the rate of molecules entering this region? Pardon? Jx times the area, because J is number per unit time per unit area. So if you multiply by the area, we get the number per unit time. Okay, that's coming in from the left. Going out in the right, it's a minus because now they're going out, and it's a little bit farther over in x. And again, we have to multiply by area. Now, this is going to give us the number per unit time that are piling up. If the number of molecules in this region is piling up, then their concentration is going up. Concentration is number per unit volume. So if we want to find the number per unit time, we would say that this is going to be the change in concentration per unit time times what? We need times the volume. Because if you take concentration times volume, that would be the number of molecules per unit time. What's the volume of this? A delta X. Okay, so we're almost there. We're going to divide by A and delta X. The A's will nicely cancel. The delta X is going to come down here. And this is beginning to look again like the derivative. This is, if we brought a minus sign out, we'd get J of X plus delta X minus J of X, all with a minus sign. And we divide through by this delta X, that becomes the derivative. And remember, it's going to be a partial derivative, so this would be dj dx with a minus sign. And what's left over here, we divide through by the delta x, the A went away, changing concentration with the time. This is dc dt. And we're using partial derivatives because the concentration depends on time and space. So we have this equation relating the two quantities and this equation. What did we do last class at this point? Who remembers what we did to get an equation that is just for concentration? Yeah. Combine them. Specifically, we take this for J and we plug it in for the J there. So, we have a minus sign here and a minus sign there. They'll cancel. We come down and we get a D, and we have a space derivative here, and this is really a partial, if we think of it as another space derivative. So we're going to get d squared c dx squared equals dc dt. And this is the diffusion equation. And as I promised, it looks just like that heat equation if you use temperature. Okay? They obey the same physical laws. Sometimes this is called Fick's second law, but we'll usually just call it the diffusion equation. Let's see if our units work. We said that D had units meters squared per second. Well, concentrations on both sides. Here is a two space derivatives means you have meters 1 over meters squared. A time derivative means you have 1 over seconds. When you bring it to the other side, you get seconds over meters squared, and D is meters squared over seconds. The units work. Everything works out just fine. All right. We kind of went through that a little faster because it's very similar to what we did last class. Now, let me see if I can give you a little insight into why things behave the way they do. Let's look at concentration versus position. Suppose the concentration fell linearly from a high value over here to a medium value to a low value. How is the concentration changing with time in that case? How would concentration change with time if at one instance it fell linearly? Well, what's the second derivative of a linear function? 0. So if it doesn't change with, what it means is you get a 0 for the second derivative, it means the concentration is not changing at time at all. What's happening is if you look at a little spot here in the tissue, a little value of x, you're going to have a current coming in from the left. You're going to have exactly the same current going in from the right. So it doesn't mean that there's no molecules moving around or diffusing. What it means is there's no net accumulation in the center when it varies linearly. That's why it's important to realize it's a second derivative, not a first. Okay, what happens if we have a case like this, where you have it sort of a parabola shape? High concentration on both ends and low in the middle. If you look here in the middle, are you going to have molecules piling up, or are you going to have them disappearing, or are you just going to have a constant concentration? What would you have in this case? Well, if this is like a parabola and you took two derivatives, what would you get? If this was x squared, you get a constant. You get just a constant too. You wouldn't get 0. Which means you would have molecules piling up. You have some positive constant, because this is a positive second derivative. So what you'd have is you'd have molecules going down their concentration gradient. So they're coming in from this side, and they're going down their concentration gradient. They're coming in from this side, and they're piling up in here, and you end up getting a positive time derivative. So it's really telling you that if you have coming in from both sides, you're piling up in the middle. And similarly, if your concentration gradient looks like this, it's now an upside down parabola. And now you'd have, if you looked at a little spot in the center, you'd have molecules leaving both sides if they're going down. And then you'd have molecules disappearing. Well, the second derivative of this, it's like a minus x squared, would give you a minus a constant. So you have a negative time derivative, concentrations going down. So it's kind of important to be able to interpret an equation like this in terms of what's actually happening in the physical world. Okay, questions on that so far? Okay. The diffusion equation is one of the big 3 partial differential equations of physics. There's the Laplace's equation, there's the wave equation, and there's the diffusion equation, are your standard partial differential equations. So we're looking at one of the important ones. Alrighty. I'm going to now show you the most famous solution to the diffusion equation. And we'll analyze it. Okay. I'm not going to ask you to derive this thing. In fact, I don't quite know how this was derived. I imagine somebody with hundreds of sheets of scrap paper, trying various things till they got it. But let's assume now we're going to deal with one dimension. So let's say you have a pipe and you're going to have particles diffusing in the pipe. This is a very long pipe, so we don't have to worry about the ends. It starts with nothing, and then at time 0, you inject a bunch of particles right here at x equals to 0. So you actually inject n molecules per unit area, because if you made this pipe bigger cross-sectional area, you'd have to inject more molecules to have the same concentration. So at time equals 0, you inject all these particles right at the center. And then you watch them diffuse, and they're going to diffuse out through the pipe. And what we want to do is we want to try to predict what the concentration is in function of time and space. All right, I'll tell you the solution. We're going to basically do guess and check, where this would be a very hard guess. But the numbers, proportional to how many you put in, you're going to get a 1 over 4 pi dt square root. And then you're going to get an exponential minus x squared over 4 dt. Okay. Anybody know what this function exponential, the minus x squared, looks like? It's a normal distribution or a bell-shaped curve. It goes up to a peak at the center and it falls off on both sides. And we're going to look at that some more. It's a little easier for working analytically if I don't want the t down here. I'm going to take my constant factors out. And then I'm going to write this as, this is t under the square root in the denominator. That's really t to the minus 1/2 times e to the minus x squared over 4 dt. Now I claim this obeys the diffusion equation. What I want to do now is I want to verify that. So how are we going to verify it? What do? We just plug this quantity into the diffusion equation and see if it works. We don't need to worry about the constant factor because it's going to be the same on both sides and will cancel. So what we need to do is we need to take some derivatives. So let's go through and just verify that this actually works. And we'll start with dc dt. So we're going to ignore this constant factor because we know it doesn't matter. We just have to take this time derivative. Okay, now we got a little bit of a problem in that this has time here and it has time here. We need to use our product rule. So what's the product rule? Anybody remember?
First term times the derivative of the second term, the derivative of the first term times the second term.
Okay. We take the derivative of that times that plus that times the derivative of this. So what's the derivative of t to the minus 1/2?
Negative 1/2
T to the what?
to the
Negative.
negative 1/2
Minus what? So minus 3/2. So we get this times this. And then we had the product rule, we get plus t to the minus 1/2 times, and now we got the derivative of this, and now we still have a problem because we need to use the chain rule. The derivative of an exponential is just an exponential, so we get our exponential back, but the derivative of the inside is kind of complicated because you got 1 over t. So we got to take the derivative of this whole argument, which is going to give us a minus x squared over 4d. And what's the derivative of 1 over t? Minus 1 over t squared. Okay, so it looks complicated, but we can factor out, let's factor out the exponential itself, because it's in both terms. And here's a 1 over square root of t, and this really has a 1 over square root of t times t in the denominator. So we'll take out 1 over square root of t. And what are we left with? Well, this term is going to give us a minus 1 over 2. And if you took out the square root to the minus a half, you're going to be left with a 1 over t. This term is going to give us a minus and a minus or a plus x squared over 4dt squared. So we get that somewhat complicated thing, but not too bad.
And do we have to multiply by the constant and over?
If you wanted to, yeah, so I kind of said that we were going to ignore this, but this you technically have that constant in front of all these terms, and it would cancel out in the end. So let's just agree to make life easier on ourselves and ignore it. Cancel it out right from the start. Okay, so we now got this time derivative. Now what we want to do is we want to check the second spatial derivative and see if we get the same thing. The good news is the spatial derivative is easier because it's up here, but it's not down there. But the bad news is we got to do two derivatives. So let's try it. dc dx equals, okay, we're ignoring the constant. This is the partial derivative. If you're taking derivative with respect to x, t acts like a constant. So it's just going to go t to the minus a half. And we got the same thing. We got the derivative of the exponential. We got to use the chain rule. The derivative of an exponential is the exponential. And then we got to take the derivative of the argument, and that's going to be a minus 4 dt x squared, the derivative of x squared, 2x. Okay, so that was easier. But like I said, let's go ahead and simplify this a little bit. We'll take the minus, we'll cancel out the 2 and the 4. So this is going to be x over 2d. And then this is a 1 over t, and here's the t to the minus a half. So that's really a t to the minus 3 halves times the exponential. And now, that was easier, but now the bad news is we got to take another derivative, and now we got the product rule, x and e to the minus x squared. So we're almost there. We just got to take this second derivative. So we take the derivative of the first derivative. So we're taking the derivative of this. We got minus t to the minus 3 halves over 2d. And we have now x times the exponential. Product rule. What did we get for our first term?
X.
We're just going to get, we're going to get, if we have x times the exponential, first we'll take the derivative of x.
Okay, so just x.
So you just get the exponential. And then the second term is going to give us plus x. It's plus because I already factored out that minus sign. And we take the derivative of the exponential, which is a chain rule. So what we're doing is practicing product rule, chain rule. The derivative, that gives us the exponential back, because the derivative of the exponential is the exponential plus the derivative of the thing inside, which is minus 2x over 4 dt. So we will now see what we get here. We get minus, this is 2d, and then this, I'm going to write 3 halves down below. We're going to get the exponential, and then we're going to get 1 plus, minus 2x squared, but the 2 will cancel the 4, so x squared over 2dt. Now, the claim is that d times this is going to give us this. Well, let's see if that's true. If we multiply this second derivative times d, it'll kill off that d. We're going to have a minus sign out front. Well, we have a minus sign inside here, so we'll have to take the minus out front. Here you got a t to the 3 halves and a 2. Well, here you have a 2, here's a t and a square root of t, so it's really t to the 3 halves. Here you have a minus. We had a minus out front, so it's a plus x squared over 2, and there's a 2, 4. One of these d's cancel with that one, so you're left with a d, and then you have a t, which is related to the 5 halves. Well, there's t squared plus a square root. That's related to the 5 halves. So what we find is that if you take this and you multiply it by d, it's exactly that. It works. Almost by miracle. It's a complicated mess, but it obeys the diffusion equation. So I wanted to just go through and verify that because, like I said, it's not at all obvious how you know to do that in advance, but it's amazing that it works. Let's see what it looks like as a function of time. You all told me that this is going to be c at different times. You said this is a bell-shaped curve. So it's got this general shape like that. And if you wanted to figure out how wide this was, well, let's figure out how far out you have to go. And it's got some peak. But then how far out do you have to go in order for that peak to fall off by 1 over e? So basically 1/3 roughly of its value. So if this is the peak, then 1 over e is going to be about here. So what value of x is that? Well, that is when e is to the minus 1. E to the minus 1 is 1 over e. So that means you got to have x squared over 4 dt equal to 1, which means x is 4 dt square root. So this point is going to be 4 dt square root, where it's roughly fallen off. That means that as time gets longer, this thing spreads out more. But it spreads out not as linearly with time, it spreads out as the square root of time. So initially it spreads out pretty rapidly, but then that square root kind of flattens out as time goes by and it spreads out more slowly. So at very short times, right after you inject the particles, you're going to get a very narrow Gaussian. But you're also going to get a very tall Gaussian, because at very short times, you're going to get a very tall function, because this is going to be really small. It's in the denominator. So at short times, it looks like this. So short, I'll call this medium times. At very long times, it spreads out even more, but it goes down. So at very long times, it's beginning to look like this. Almost flat, but eventually it'll spread down. So that's how the diffusion equation behaves. Things spread out, and they get spread out with time. Their high concentrations drop quickly, their low concentrations slowly build up, and we know exactly how to describe that as a function of time. Now, there's a few things about this that are interesting. For instance, suppose you wanted to calculate the total number of particles we put in. Well, you ought to be able to take the concentration times the volume and get the number of particles. But since the concentration is changing with time, you really have to integrate the concentration over the volume to get the number of particles. So we're going to look at integral from minus infinity to infinity of c of xt dx. Okay, well that's going to be, this is what our c is going to be. This is the number, this is the integral over x, so it's number over 4 pi dt times the integral from minus infinity to infinity of e to the minus x squared over 4 dt dx. Now, it would be fun to spend some time evaluating this integral. It's analyzed in Appendix K of your textbook. But I don't want to spend a lot of time working on that integral, so I'll just tell you what it's equal to. This integral equals 4 pi dt, which is exactly going to cancel this, and you end up just getting n particles, technically particles per unit area, since we really didn't we multiply. If we wanted the total number, we have to have an A, so we get it over the volume. So what it says is that the total number of particles conserved in this diffusion. This is an example of diffusion. If the number of particles are conserved at the base of the diffusion equation, the total number doesn't change, they just spread out. Okay, let's look at another case. Running out of board space. So let me look right here. Suppose you wanted to get the mean value of x. Where's the average value for these particles? Well, to get that, you would do integral minus infinity to infinity of x, c of xt dx. Technically, you divide it by your n, your total number of particles. Okay, and this is this complicated function. Did anybody tell me what this integral goes to? This one, I'll give you a hint. You don't actually have to do any calculations. You make just sort of general argument. Well, our concentration is an even function, but x is an odd function, which means that if you integrate it over all space, an even times an odd is odd. You're integrating an odd function over all space, it's just going to go to 0, just because anything that happens over on this side is going to cancel out my hand saying over on that side. So just automatically, this average value is 0, which just, you can see it right here. The peak of this curve is the average is going to be right at the peak, and it just drops down, but it doesn't. The peak doesn't shift. If the peak shifted, we'd be looking at diffusion with drift, which is another problem. But pure diffusion, the average value just falls down. But one more we want to look at. What if you tried to look at the variance, x squared, and you take that average value? That's going to be x squared c of x t dx over n. And now you can't use that trick. X squared's even, c is even. You actually have to do the integral. Again, I'm not going to stop and do this integral, x squared times the bell-shaped curve. I'm just going to tell you the result that you get. It's pretty clear that it's going to be increasing with time because this thing's spreading out. So if you just jump to the result, 2 dt. So in other words, the RMS distance, which is like the standard deviation of the root mean square, which is the square root of the variance, is 2 dt. And it's almost identical to this, except for just this constant factor. What that means is this wasn't, it spreads out the same. This just isn't technically the root mean square distance. That's the 1 over e distance. But the important thing, this distance goes a square root of time. So in general, diffusion happens as a square root of time. It's very effective at very short distances. Very short distances, diffusion happens very quickly. As you get to longer distances, diffusion gets much more slow. And you can see that all from this, this example. Questions on this? It's a lot of math, a lot of taking derivatives, but I really wanted you to see that this solution really works. Okay. Let us do one more case where I won't do any math at all. I'll just draw pictures and we'll see qualitatively what happens. Suppose you have the same cylinder, but instead of putting a bunch of particles at the origin at time t equals 0, you take it and now it's only a finite length and there's a wall here. And this side has a concentration C0 and this side has nothing. And at time 0 you remove that wall and you're left with two particles diffuse amongst each other. What does the concentration look like as a function of time? Well, here's concentration of x, here's C0, and initially, right before you remove the barrier, it's a step function. That's at t equals 0, basically, or immediately afterwards. Now, a little while later, what happens? Well, out here, this was pretty flat, so its second derivative was pretty zero, so it wasn't changing much. So nothing changes much except right here near the boundary where it begins to diffuse. So for short times, you mainly get just a diffusion over a very small distance. But as time goes by, you begin to get this diffusion to be going over longer distances. That's T medium. Until finally, what's it going to be after a long time? It's going to be half of its original value. And all the particles are going to spread out. It's a finite length tube now. All the particles will spread out, so half of them will go over here, half of them will remain over there, and it's going to be half of its original value. So you can kind of see the diffusion happening. You can actually define this. There are mathematical functions called error functions which would describe this exact type of diffusion, but we won't be looking at the mathematical part. But this is for T long. It's the asymptotically long when it reaches steady state. But it takes a long time to reach this state, whereas it takes almost no time to get this initial effect. Alrighty. Now, questions on that? Okay. We're going to look at another example of steady state diffusion. And let's see here. This is the problem 4.37 from the book. So let's see what problem 4.37 is. It's almost problem 4.37. We're going to make a slight change. The goal of this problem is to estimate how large a living cell in oxygenated medium can be before it is limited by oxygen transport. So how big can you make a cell? So that's kind of a fundamental question that we're going to ask. So let's see what physics says. Assume the extracellular space is well stirred with a uniform concentration C0. Now the problem says a sphere of radius r. We're going to again do a one-dimensional problem. So we have an animal that's just a slab of thickness L or 2L, minus L to L. Inside the cell, oxygenation is consumed at a rate Q molecules per meters cubed per second, and the diffusion constant is D. So let us see. We now have a source of molecules. That is, or really a sink, because oxygen gets used up in metabolism, so oxygen molecules will disappear and be turned into heat and carbon dioxide and things. So if we're looking at just oxygen, we really have inside the tissue a sink, which we'll call Q, which has units of molecules, and this is really the molecules destroyed per meters cubed per second. So our diffusion equation picks up an extra term. If you now have a source, you have an extra term in the diffusion equation, and it's really going to be a minus Q. It's minus because they're destroyed, and you get an extra term in the diffusion equation. Well, that's bad news because it makes it more complicated. But the good news is it said it was steady state. What's steady state mean? No change with time. So that thing goes to zero. Now we can just use total derivatives instead of partial derivatives. Bring the Q over the other side, divide by D. Our basic equation is D squared C dx squared equals Q over D. Now, if you're thinking to yourself, this looks a lot like that heat problem we saw in last class, it is. Okay, it is. So what you have is the concentration. It's a function of x versus x. Here's L. Here's minus L. This is the tissue. And has a diffusion constant D. And we said it's well stirred, so C0 outside on each side. And we're going to have a boundary condition in that the concentration right inside the cell is the same as the concentration outside. In other words, the membrane doesn't act as a significant barrier to the diffusion of oxygen. So we want to know how big can this cell be. So we're going to do the same guessing check. Now, by the way, sometimes I make mistakes. If you see any mistakes, please remind me so we don't all get confused. Okay, here's your guess. What did we use for our guess last class when we had an equation like this? We need a function that you take two derivatives and you get a constant.
AX squared plus B.
We used a constant we don't know, x squared. We didn't use a b times x because we know this is going to be even symmetry, but we had a new constant. Well, I'll call this one b. I don't want to use c because that's the concentration gradient. I don't want to use d because that's the diffusion constant. I'm running out of, so I just call it b. Okay, what do we do with this? We stick that into our equation, and we see what we learn. Two derivatives of ax squared
2A.
gives you 2a. This term doesn't give me anything. Equals q over d. So we immediately get that a is equal to q over 2d. So we've already found out what one of the constants is. Okay, now what do we do next? We have to have the concentration be equal to C0 at x equals to a. So how do we implement that? What do? We just set this thing, concentration, which is A, and A is Q over 2D, at x equals to L, so x is going to be L squared, plus B equals C0. That's really just setting the concentration at the boundary equal to the concentration outside. And that gives us B equals C0 minus Q over 2D L squared. So we now have A and B. And what we end up getting then for the concentration, just a function of space because it's steady state, we're going to have a C0. We're going to have this term A, which is positive, as a Q over 2D. This term in B, which is negative, as a Q over 2D. So we're going to factor out a minus sign and a Q over 2D, and you get L squared minus x squared. So the x squared term is positive when you do with minus signs. And that's our concentration. Looks a lot like last class. If we plot it, L minus L, here is C0, here's the concentration outside, It's a parabola. It's actually a positive upright parabola. Minus and minus gives you plus. At x equals to L, it goes to 0, so it's continuous. It looks something like this, where the value at the center is just setting x equals to 0. You get C0 minus Q over 2D L squared. So we can calculate the entire oxygen distribution. Now, here's where it changes up a little bit from last class. You know, oxygen can't go negative. You can't have a negative concentration of oxygen molecules, which means that if this quantity is big enough, this will dip down and reach all the way to 0, and it can't go any farther. If it goes any farther, our model's wrong. Basically, if it goes any farther, the tissue at the very center has no oxygen, it dies, can't produce metabolism, so Q goes to 0. And the whole model breaks down if oxygen goes to 0. In fact, it might break down if it gets close to 0. So, what that means is really, this quantity has to be positive. Greater than 0. Well, if you move things around and you figure out what that means for L, L is going to have to be less than C0, 2D, over Q square root. That's really a condition on the size of the cell. If the cell gets any bigger, then this concentration will go down to 0 and it'll have a dead core. And this is basically the cell will die. So this is our limit on how big cells can get. Okay? So we can actually put some numbers in here. In the problem, it says the concentration is going to be, and in this problem uses moles, won't matter, 8 moles per meter cubed. Okay, that's not exactly the concentration of oxygen in your blood, but it's on that order of magnitude. So it's kind of the oxygen concentration that your cells see. Diffusion constant is 2 times 10 to the minus 9 meters squared per second. This is the diffusion constant for oxygen diffusing in water. It's just what you get if you measure it. It's a small number, oxygen doesn't diffuse all that great. And Q, well, Q depends on the animal. We're going to find 0.1 mole per meters cubed second. This is actually a number for bacteria, so it might be a little high for a mammal, but it's for an active growing cell. So if we put all these numbers in, you're going to get L has to be less than 8 mole per meters cubed, and a 2, and the D is 2 times 10 to the minus 9 meters squared per second, divided by 0.1 mole per meters cubed second, all under the square root. Okay, the moles or molecules cancel. That's why I said it didn't really matter whether you're using it in moles or not. Let's see what the units, if the units work, first of all. The 1 over second cancels the 1 over second. Meters cubed here cancels that meters cubed. You got meters squared, but it's all under the square root. Units work. If you do the calculation, what you get is 0.57 millimeters. Convert it to millimeters. So it's about 1 millimeter total size for the cell. You get bigger than that, you just can't have diffusion diffuse in. And that's actually a little bit of an overestimate because you really don't want the oxygen even getting close to 0 at the very center. So cells are limited to be sort of hundreds of microns, a couple hundred microns. That's what we saw those paramecium and the amoeba in the first day of class. We saw them were that size. Well, that's because they're oxygen limited. And the reason we can exist is we have lots of cells, but more importantly, we have a circulatory system which carries the oxygen through convection to all our cells. If we didn't have the circulatory system, we'd be dead. So really what we're saying is how big can a cell be before it has to develop some, or how big can an animal be before it has to develop some circulatory system is basically this size. Okay. So that is how big cells can be. And it's interesting, I think, to see that basically the way animals and cells evolve is, over time, is limited by basic physical effects like this. Now you might say, is it oxygen diffusion that actually limits the size of cells? Well, you can look at other things. You can look at diffusion of carbon dioxide, waste products, and there's a lot of other things that could be, but actually oxygen diffusion is one of the main factors that limits how big you can make a cell. All right, so this is a good example to show you solving the steady-state diffusion equation. It's also a good example to show you how you can use this to actually calculate something important in biology. All righty. Any questions on that before it all goes away? Now, let's do something that I know you guys will all be interested in. That is, let's look at some of these homework problems and see if I can give you a few hints on how to get started. Okay, homework problem 8. Homework problem 8 is one of these problems where I'm really asking you to calculate concentration. So it's not too much about the fusion itself. It wants to know the concentration. Sometimes we write concentrations as a bracket of oxygen in air. And really it's asking you to use the ideal gas law. PV equals nRT, where for chemists this is number of moles, that's the gas law. Oxygen, you can get N divided by V is basically a concentration. So you can, all you got to really do is take typical for our air, typical pressure, typical temperature, work it out, and you can calculate the concentration of oxygen in the air. And I think they may also ask you for the concentration in water. Anyways, it's just calculating concentrations. Well, it wants the concentration of liquid water, of the water itself, not the oxygen in it. That you've already done in the previous problem, so it's almost a repeat. So that's problem 8 in the assignment. Problem 12. Problem 12 is looking at something similar to what we talked with Boltzmann's equation, but it also relates to diffusion. If you take some small little particles, say some little pollen grains or something, and you let them suspend in water, and then you just sit there and you wait until they settle down to a steady state distribution, they're basically going to form some steady state. They're not all going to fall to the bottom because there's thermal motion. Some of them will be floating around in top. And you're going to get essentially a Boltzmann factor of distribution. So it looks like where this is going to be the height, that's the energy, that's the thermal energy, the gravitational potential energy. It looks like a Boltzmann problem. What's it doing in chapter 4? Well, it's really a steady state diffusion problem solution too. You're given some data. You're given enough data that you can basically find, this can be right, this is minus z over some length constant. And you're given some data that would allow you, if you plot it, to work out what the length constant is. You plot it on semi-log paper, z versus concentration, which goes to semi-log. And you should be able to work out what the length constant is for this case. And this is historically important because a Frenchman named Perrin made this measurement. At the time he made it, people knew what the gas constant was, and they knew what Boltzmann's constant was. And if you remember, Boltzmann's constant and the gas constant are related by Avogadro's number, but they didn't really know a good value for Avogadro's number. So when Perrin made this measurement, by measuring the distribution and getting this length constant, he was then able to go put in the temperature, get Boltzmann's constant, and by getting Boltzmann's constant and he knew the gas constant, he was the first person to measure Avogadro's number by this technique. So that problem's got a little bit of history behind it. You can go through and you can try to see how you can calculate the Boltzmann's constant using data very similar to his. Okay. Problem 20. Problem 20 develops something called the Lewis number. And the Lewis number is just defined as the diffusion constant of molecules over the diffusion constant for heat. We talked today about the diffusion constant for molecules, and we used the value for oxygen. Last class, we talked about the thermal conductivity and the heat capacity. We put them all together and got a diffusion constant for heat. Both diffusion constants have the same units. You take the ratio, you get a dimensionless number, which is called the Lewis number. Now, what's this tell you? It tells you which happens faster. Do molecules diffuse faster or does heat diffuse faster? And it has some relevance. Remember when we talked about the dolphin and we were trying to see if you could somehow make a way where the dolphin could be able to breathe water through gills and still be a warm-blooded animal? Well, if you could make the diffusion of heat really slow and the diffusion of oxygen really fast, you can maybe make a system where they could get the oxygen without losing all the heat, and it might work. But that requires a certain Lewis number. And when you analyze these values, and the problem gives you some values for these properties, you'll find that it doesn't really work because the Lewis number means, the Lewis number is big basically, which means diffusion of oxygen, the Lewis number is small. So diffusion of oxygen is kind of slow compared to diffusion of heat. So if you want to try to get oxygen from some system, you're going to get heat too, for almost any case. So the Lewis number is developed in problem 20. You just asked to put in some numbers, do some estimates to try to see what the diffusion, the Lewis number is in various cases. Okay. Let's see, we still got a little time. Problem 22. That's the next problem in the homework. We talked on the very first day of class about alveoli, which are the little terminal air sacs in the lungs. So you come down here, and these things are about 250 microns in size, and this is all air in here. And why do we even have lungs? We have lungs so our blood can pick up oxygen. So there's capillaries that go around here, carrying blood cells. So what's going to happen is oxygen in the air has to diffuse over to the edge of the alveolus and then diffuse in to the center of the capillary and then get attached to the red blood cell. Now the question is, which happens fastest? Does oxygen diffuse basically through the alveolus, let's say from the center to the edge faster, or does it diffuse through this little region in the capillary faster? And you might say, well, it's going to be fastest in the air, because the air has a much higher diffusion, or has a much higher diffusion constant, and it does. But someone else might say, well, it's going to be faster through the blood, because this is a tiny little distance compared to this great big distance. And I probably exaggerated this. This is 250, and this is more like 10, so I probably should have made the capillary even smaller. So you're going to calculate which one is really the limiting factor, is diffusion. And you're going to use this idea of x equals 2dt square root. This is a relationship between space and time. So if you want to find how long it takes to diffuse, say, through the capillary, you'd solve for time and you'd use x as maybe the radius of the capillary, and you'd use the appropriate diffusion constant. And if you wanted to find the time to get, so you go from the center to the edge here, you'd solve for t, and you use this radius, and you use the diffusion constant for air. And technically, you probably would want to do a 3D problem for the alveolus because it's more spherical, but we can ignore that. You can just go ahead and use the 1D relationship. The only difference between 1D and 3D is this 2 changes to, I think, a 6 in 3D. But you can just go ahead and pretend it's all 1D. All right. Here's another problem. This equation is going to be central in several problems. So let's look at problem 23. Okay, why do we breathe? Why don't, instead of breathing, why don't I just stand here like this and let oxygen diffuse down into my lungs? Okay? Would that work? It sure be a lot easier to go than to have to go. So, does it work? Well, you can calculate how long it would take for oxygen to diffuse down into my lungs. You can solve this for T. You can figure out the diffusion constant. And x is going to be basically roughly the distance from my mouth to my lungs, which you can estimate. And you can estimate this time. And if that's a real short time, if that's a microsecond, then this breathing is nonsense. We ought to just stand there. And if this is 3 hours, then, oh boy, we better breathe, otherwise we're dead. And you can probably guess what the answer is going to be. But you can actually calculate this in problem 23 from some simple estimations in this expression. So that's what you're going to be doing in problem 23, why we bother to breathe. Okay, problem 24 is going to be a similar one. But instead of talking about breathing, we're going to talk about nerves. Now I got to teach you a little bit of nerve physiology. Here's a nerve axon. A nerve cell, like a long cylinder, and at the end it kind of bulges out like this. So this is a nerve, and this is a muscle cell. And at the junction between the nerve and the muscle is what's called a synapse. And they aren't directly electrically coupled to each other. What happens is a nerve fires an action potential. We'll be talking about all this stuff in chapter 6. The nerve fires an action potential. It gets to the end of the nerve, and the nerve releases a chemical called acetylcholine. C-H-A-C-H, I think usually, yeah. And the acetylcholine diffuses across this junction, and then hits receptors on the muscle, and that causes the muscle to fire an action potential. So it's the chemical synapse between the two. And what we really want to know is how long does it take for this diffusion to occur? And the real question is that the limiting factor? Because it takes like a millisecond to make this transition, to send the signal across from nerve to muscle. And is that explained by the diffusion time, or is it explained by something else? Well, the problem gives you the width of this gap. So you can basically, it gives you the diffusion constant, you basically solve this equation for time and calculate the time and find out how long that takes for the diffusion to happen. And then you can ask yourself whether it's the limiting factor or not for diffusion at the nerve muscle synapse. So it's kind of a simple problem in diffusion wrapped up in an interesting problem in physiology. So I'll let you do the calculation and figure out what the answer is. Okay, 2 more. We still got a couple minutes. 41's a little bit more solving the steady state diffusion equation. So we solved, you're going to notice, always watch for steady state. Because if it says it's in steady state, you know that this is either 0, or if there's some source term, which there is, you would get a Q here. And what we're going to be doing is you're going to be solving problem 41. It's going to be very similar to what we did in class, except it's going to be for the cornea of the eye. The cornea of the eye must be transparent, so it contains no blood vessels. So if there's no blood vessels, you basically you don't have any oxygen coming in, but oxygen is getting used up because it's still doing metabolism. So it's going to obey an equation like this, but there's no blood vessel. So it's going to look like almost one big cell then. And you're going to have to apply various boundary conditions to a model. So you're going to make a 1D model similar to what we did before, and you're going to have to solve this equation. You're going to guess a parabolic. And the only issue is the boundary conditions. In one part, it has boundary conditions where you have the same concentration on both sides. And then there's another part where it says that the outer area, you have your eyes closed, so you have a low concentration of oxygen right in front of your eye because it's not exposed to the outside. So one side's here and one side's here. And then it has another case that you have an oxygen impermeable contact lens. So that means you have a lens over one end. So you have basically J is equal to 0. You can have no oxygen flowing out. And J, remember, was equal to minus d dc dx. So now you have a problem where you don't know what the concentration is on this end, but you have a concentration derivative has to go down to 0. So it's dc dx equal to 0. Okay, one hint to remember, with this problem, it goes from like 0 to L, and it's different on the two ends. You can't throw out the linear term because you don't know that it's symmetric. It's not an even function anymore. So you'll have to, it takes a little bit of solving, but it's basically like what we did in class. The boundary conditions are a little bit different. If you have problems, bring the problems, and we'll talk about them in class. Okay, the very last problem, 42, is what did we get in class? In class, we found that the size of the biggest cell was dc over q. Well, that depends on the diffusion constant for oxygen. And oxygen diffuses much better in air than it does in water. So if you change the diffusion constant, it's going to make some change into the maximum size of your cell, or the maximum size of your diffusion distance. And it turns out insects don't have blood like we have, they have little air tubes that go into their body and deliver oxygen to their cells, rather than tubes filled with water carrying blood. So the problem asks you to just basically analyze how diffusion distances change for insects as opposed to other cells that depend on diffusion within water. So it's kind of an interesting problem in comparative physiology. And that is your 8 problems. Hopefully you can get a start on them this weekend so that you can bring questions Monday, or if not Monday, definitely Wednesday, so that we're going to have it done on Friday. Okay, so that's it for today. And I will see you on Monday. If you haven't gotten your exam or homeworks back, you can come get them from me.